Our next presenter is from Andelyn Biosciences. He is Matt Niloff, Chief Commercial Officer. Thanks for joining us today. Welcome, Matt.
Thank you, Perry, and thank you everyone for joining. So today I'm going to talk about our AAV Curator platform, enabling gene therapy program success through optimization by design. This is a platform that we've had for quite some time. We newly branded it, so this might be a new term for you from our company. I'll go through a little bit about our introduction, overview of our capabilities, and then we'll kind of do a deep dive into the Curator platform, looking at the cell line, some key considerations. What does optimization by design really mean? And then what is the impact for reducing cost of goods? We'll look at what does partnering with us look like, and then how to get started working with us. So I usually like to start with talking a little bit about our more recent partnerships and collaborations and awards. I think it really just showcases how our know-how is really being adopted and considered very strongly by the industry. Of note, we're doing a lot of late-stage work. We signed with Grace Science. This is a phase 1 all the way through phase 3. This was a full tech transfer. We're working with the Bespoke Gene Therapy Consortium with the FNIH and CERM. We signed a deal with Armatis for the full CMT1A program from development all the way to GMP manufacturing. Very exciting for us is we're working with Ultragenyx doing PPQs. So very exciting for our facility that came online several years ago to now being, making a commercial material. Very exciting. And then other partners, OHU, PureSpring, and Odilia. And these are just the ones that we've been public with. I like to show our history. I think it really showcases, where our know-how comes from. Basically, we've been working with AAV since the early 90s out of Nationwide Children's Hospital. We developed a viral core back in the late 90s and then established a GMP facility working on muscular Duchenne's dystrophy with the first patient dose there. We then developed, back in the late 2009, 2010, our own HEK Curator adherent cell line. We were instrumentally involved in the development and early work and clinical production of what is today's Zolgensma. We then built out a second facility for clinical manufacturing. We doubled our capacity. In 2020, we realized that our clientele really wanted to have the full pathway from development all the way through commercial manufacturing. And so we spun out and formed Andelyn back in 2020, built our facility. Through COVID, we had a strategic investment from Danaher or Cytiva, and then continued to bolster our capabilities with the suspension cell line and scaling all the way up to 2,000 liters. We had our official grand opening about a year ago. And as I mentioned, we're actively doing PPQs today, and we've touched upon a great number of diseases in the rare disease space from NCH and our work here today at Andelyn as well. So interestingly, there are 6 AAVs on the market today, and we've done the development work and some clinical work for 2 of them. So I mentioned Zolgensma. This came out of Nationwide Children's Hospital with our SMEs developing that process, and then Novartis ultimately acquired it. This was for SMA type 1. And then also the original process for Elevidys for Duchenne muscular dystrophy, again, we worked on, came out of Nationwide. Again, it's really important to show that we've been involved in the AAV space for quite some time and understand quite a bit of the nuances to get a drug from development to manufacturing. So, with that coming out of Nationwide, we've got a tremendous amount of knowledge from, development all the way through to the clinic. Our team from the early days of AAV clinical trials were involved. We've been in the dosing rooms. We've helped, do work for the toxicology studies, drafted and reviewed clinical manual of operation, identified instrument administration materials for for compatibility with the vector, and just kind of work through and through to really determine what are the key considerations for development that impact manufacturing. To date, again, we've been at this manufacturing well over 20 years, over 450 sublots, GMP sublots, supported over 75 INDs. We have combined about 250,000 square feet of total space. We do have a vision of turning hope into reality. It's our clients that have the innovative therapies, and so it's our mission to accelerate those therapies and bring as many of them as possible to more patients. The work with SMA type 1 and Sarepta's early process, the two pioneering patients are actually in our names. So Andrew and Evelyn, combine those names and you get Andelyn. So a little bit about our history there. In terms of our full capabilities, we are an end-to-end partner. We feel this is very important for managing efficient program management. We can produce plasmids. We do plasmids engineering, cloning services, full development of vector manufacturing, preclinical talks through GMP and commercial, all the way to 2,000 liters. And then we do have automated fill finish capabilities as well. We have the full gamut of analytical development and quality control. We're doing about 90% of our assays in-house. We've got a full team for quality assurance, offer regulatory support, and we have a number of ways of working with our partners in a flexible manner. So we do operate out of 3 facilities. On the left is our development facility. This is where we do process development, analytical development, preclinical manufacturing, tox runs, though tox runs can be done also at our manufacturing site. In the middle, we do have our commercial facility. It's about 180,000 square feet, total space. And we do have suspension capabilities from 50 liters all the way to 2,000 liters. And then we do substantial adherent work as well in 30 hyperstack or 60 hyperstack sublots, full QC control throughout, supply chain, warehouse, and office spaces located here as well. And then for the plasmid work that we do, it's out of a space at Nationwide Children's Hospital, about 9,500 square feet there. This is just a view of our technology platform. We have standardized on the Pol/Cytiva STRs. We do remain technology neutral. We are doing tech transfers as well. If there is a process in which the client or through discussion with us from a regulatory standpoint, we really don't want to change, then we will certainly be open about bringing in the technology so as not to change the process. But basically, full capabilities, to produce the plasmids, the materials to start, all the way through upstream, both suspension and adherent, hyperstacks, isellus. All of the technologies are single use, depth filtration, and then ultracentrifugation, chromatography, sterile filtration, and filling, all of the release testing machinery necessary as well. Okay, so just to get into the AAV Curator platform, this is both an adherent and suspension modular platform process. It was established based on the ability to be configurable and ensure that the end process is really curated to a program's specific needs. The adherent process is a low serum system utilizing a fortified calf serum, no FBS, with progressive reduction through the production process. The suspension process utilizes chemically defined serum-free media for maximum flexibility across all scales and multiple bioreactor platforms. What's important for driving down lead time and startup time is we do have a standard bill of materials that includes off-the-shelf products and has a security of supply built in with full QC testing. From GMP starting materials, as I mentioned, we have our Curator HEK293 cell lines. We also offer off-the-shelf RepCap and P-Help plasmids, though of course we can work with our clients if they have those produced already. Upstream, we do certainly the necessary plasma DNA transfection, media harvest, and then a number of varying technologies for purification that will depend on what's optimal for the process. And then we do have a full library of platform assays for characterization, release testing, stability, and device administration stability. So our cell line, it's a clonal HEK293 cell line, and we've done all of the DOE work that ultimately systemically improves AAV titers for multiple serotypes. Just a little bit of data here on the right, you can see we're comparing to another external commercially available cell line showing increase in titer for various serotypes. We've also looked at optimal growth, really to drive high cell density to maximize vector production. And you can see here we find the sweet spot with the reduction of DNA per cell in conjunction with that optimal cell density to maximize AAV titer, ultimately productivity. So what's interesting is, the FDA has been talking more and more about platform technologies, and platforms are needed really to usher forward the 10,000 plus rare diseases, and that a well-defined manufacturing platform needs to be established. There was a recent exercise done with ARM. It was engaging with the FDA really to kind of look into what does that look like, and they put together a number of proposals. And so it's understood by the FDA that even though a platform is required, there are certain areas in the process that need to be adapted, right? Looking at different process parameters based on the program, looking at plasmid ratios, growth rates, and ultimately you want to be able to adapt independently each unit operation as needed for a specific program, right? And so based on the transgene, you're going to have some variability. That's really important. So it's not just having a static platform, but it's really about the methodology to be able to adapt to the transgene. So really what's concluded is platform approaches are necessary to accelerate and enable therapies, but product-specific approach is necessary. So with that, there are a number of considerations that need to be examined for a given program, right? What's the patient population? What's the indication? What's the delivery route? What is the regulatory strategy? Product life cycle and strategy? How does the product perform? What's the predictive performance? Dosing? What's the patient morphology? And then there are a number of questions to really dial into with the CDMO about their platform. Is there a scaled down model? Have they shown scalability? What is the CDMO's platform's anticipated productivity for a specific program, for a specific serotype? And how does the early work impact COGS for the product? And then ultimately, how is cost reduced per patient? And there's a certain number of key attributes that we need to be striving towards: the right scale, what's the titer that's needed, formulation, and of course the CQAs, critical quality attributes. So just taking a little deeper dive into the AAV Curator platform, as I mentioned, you need to be able to curate at each step for a specific process. And so if you look at the teal colored vector here, these are areas along the process where we can actually pull levers in order to be able to select the right approach for a given program, right? So there's a baseline platform but we are looking at various characteristics and parameters and modalities and technologies along the way, right? For TFF, pore size, what's the membrane format? As you get into downstream, chromatography, is an ultracentrifugation the right approach? Is it only columns? And then, what's the methodology for your final formulation and concentration? Are you going to TFF, dialysis? Again, a number of different technologies. And then, what volumes and what, vial sizes and types for your final fill. And then, of course, along the way, you really need to be looking at the platform analytics. You really got to understand your process, really well. The data drives everything, and that'll drive the optimization. So, through this approach, we call it optimization by design. The idea is to, achieve the right balance of high titer with quality, right? So you want to get high full to empty ratios, high purity, and have consistency in your recovery from sublot to sublot. Just a couple of case studies. Here we're looking at scalability, 2 different serotypes from small amber scale or flasks all the way up to the 50-liter bioreactor, showing that consistency, and then also another case study with another serotype from very small scale, showing consistent scale up all the way to the 2,000 liter. In this case study, the 2,000 liter was only filled to about 750 liters, and the target was 310 e to the 11. So we really stressed the 2,000 liter, and that's why you see slight reduction, but ultimately target was hit, and we're showing scalability for the platform as you scale. Just some more data and showing this consistency here with the different serotypes. So we're either using ultracentrifugation for purification or entirely using chromatography. Here we're looking at the ultracentrifugation, multiple serotypes. We've got very decent and consistent final recoveries, and then also final purity very high. Looking down here, this is of course suspension, working with either PEI or Effectivir, different scales. Looking at the target yields, good recoveries. And we generally, it's very program specific, of course, generally achieve a very high percentage full capsids. This is analyzed by AAC of 90% or higher. So in summary, for the AAV Curator platform, again, we take a very reductionist, holistic, and systemized optimization methodology approach to industrialize the gene therapy process. And really, that's what we do every day. That's why we're here. There are so many great new therapies. They're at the bench scale, principal investigators working on it, at a hospital or a biotech is working on it. And it's not really defined yet, and it may not be perfectly suitable for manufacturing. So we use our methodology and our platform to industrialize the process. It's configurable. We will assess the program with those considerations that I mentioned, and we will develop a strategy before we even start, and then we embark on the optimization process, pulling from our toolkit, doing the DOEs, upstream, downstream optimization, with the goal of maximizing program efficiency, really speeding up the time of development by doing the work upfront and utilizing this methodology. It's very data driven, right? So as I mentioned, over 20 years in AAV, we've done over 2,000 runs, including preclinical research runs all the way up through GMP. And so we leverage that data to be able to achieve high yields and high quality and just to have predictable performance. You really need to know what the outcome can be. We want to show consistency in productivity and quality along the whole process. We want to evaluate that, and we leverage the different enabling technologies that are on the market. This is a white paper exercise, just looking at what can optimization by design do for a program? So in this example, we've got a 500-liter process. It's a low-yielding vector, a 2E14 viral genomes per liter, with a target yield of 1E17, right? If you just take that process as is, in order to generate the 1E17 VGs, you need to do 500-liter upstream runs. This is a 10-week process, very expensive, lots of media, lots of labor, lots of clean room space. If you start utilizing the optimization by design approach, maybe just do the upstream DOE work, no downstream yet, you can significantly increase your titers, right? And the idea is to drive to 5E14 viral genomes per liter. Here we're now doing 2 500-liter upstreams. Okay, so we're getting 2.5 E17 VGs per 500 liter. We haven't optimized the downstream, so we're still getting about 20% recovery, and then you achieve your 1E17 VG. So we've reduced the processing time by half, and we reduced COGS by 60%. If you do further work and do the downstream optimization upfront, then you're really driving down the scale. You're achieving 2.5 E17 VGs, but you're getting 40% downstream recovery. So you can do one 500-liter run, do the purification, and get your 1 E17 VGs. So you've reduced you've reduced your processing time by 70%, and you've reduced your COGS by 80%. This has drastic impact all the way to a price or cost per patient. We've also driven high quality throughout this entire process. So what does full program partnering look like? We have a stellar program management team. We term this one team program management, direct access to all of our subject matter experts. These are our leaders. These are the leaders that have been at Nationwide from the start, and they're here to really support every single one of our customers. We do flexible contracting based on the project stage, and we live in a time now where funding is not as flowing as it was maybe 5-6 years ago, and some of our customers need some of that flexibility to start that work. Maybe you don't want to put an MSA in place. We can start work with some basic terms and conditions. We're very open, very flexible, very collaborative. We allow PIPs, so person and plant, at any time in development or in GMP manufacturing. This is a basic timeline, right? And I know a lot of CDMOs put their timelines on their websites or whatnot. Ultimately, we can develop a process and go to manufacturing and be in GMP in 6 months. We could do it, right? It really depends on the stage of the process, what it's gone through. In general, if it's the first time we're seeing it, you're looking at about a year, a little under a year. Could be more, could be less, depending on how much DOEs we decide together with the client we should do. But basically, a feasibility study 4 to 6 weeks, 3 to 4 months for your further research work, your tox production, engineering runs. This could be 1 run, this could be 2 runs, 3 runs, and then clinical production. About 5 to 6 months product release. So again, looking at about year or less. And the other thing is that if you're embarking on the upstream DOEs and the downstream optimization, we can do a lot of that work in parallel, and then the material generated can be used by us and can be used by you, our clients, to do all the assay work that is needed as well.
Matt, I'm getting the notice that we're well over time, so we're going to have to close. Yeah, my apologies for that, but you have a lot of great stuff to say. It was a great presentation. Love the story of your name behind the name of Andelyn. So thank you very much for joining us today. And any further questions that our audience has, they can contact you. So thank you very much.
Thank you.